Welcome, Mark, to the Q&A session of the seminar, and thank you for your beautiful talk. So the first question we have is, how can enterprises like this can help conservation when the real problem is environment fragmentation? Can money be better invested in land recovery? Thank you.
Yeah, so this is a really cogent question. So are we just fiddling while the landscape around us burns? Are we just looking after our own scientific curiosities while the environment degrades? So I would say that we need a joined up approach. We need an approach across several different levels and aspects and dimensions. And yes, really, the best way to conserve the natural world is to actively conserve it, to set aside land, to allow land to be rewilded, and to ensure that the societal practices that have currently got us to the state we're in don't continue. It's kind of outside the remit of the Tree of Life project to change the way the world works. So what we can hope to do is change the way the world thinks about its biodiversity. So if anybody wants to be galvanized to action, I recommend you listen to David Attenborough's recent piece on extinction. It is sobering, as is the Q report on the state of the world's plants and fungi, etc. So, but I think the way we will contribute is we'll provide, if you like, the foundations of future conservation efforts. So at the moment, maybe one tracks a particular animal species by following a few markers in its genome. And in the future, we don't know what we will need. We don't know whether or not what we really want to track is a particular locus or loci in the genome. And maybe what we do need to look at is the way the species is adapting to a changed environment, which it is forced to adapt to. So yes, we're not the total solution. I think we're part of the solution. And I hope we're a part of the solution that lasts for a long time.
Thank you.
And Mark, can I just follow up on that? It's not one of the questions that's been asked, but what are the public engagement activities that you have? Because you are part of that conversation. You're trying to impact people's way of thinking. So I guess briefly, is there a public engagement aspect of this activity?
Very much so. So we work with public engagement teams that all are, for the Darwin Tree of Life project, for example, public engagement teams at all the participating partners. And there's two sorts of things we're doing. Some of them are collective engagement, so developing ways of engaging with the public who are stakeholders in biodiversity to understand what they value about their wild environment or the species they share this space with, and helping helping us think about how we can use these genomes. And then also each partner has its own outreach and engagement activities. So for example, a very common way of the biodiversity organizations in engaging people with the concept of local biodiversity is to do something called the BioBlitz, which is where you get lots of experts and lots of stakeholders, members of the public, to come and visit a particular site of particular interest and identify as many species as you can in one go, in one 24-hour period is the usual thing that's taken. And it's a great way of doing an inventory on a site. But we hope that by linking that up with what we're doing, which is in Britain, trying to sequence all the species on the site, we can actually also immediately link people's discoveries into the genomics that we're doing. And again, have that conversation about what is the utility of what we're doing.
Mm-hmm. Great. And so the next question from the live feed is, much of Earth's biodiversity is in developing countries, which may be less able to resource this type of effort. How can the community in the Tree of Life best work with them?
Yeah, again, another great question. So my colleagues in the Earth Biogenome Project don't like me saying this, but if you take 1.5 million species and take away the 60,000 that are in Britain and Ireland, you're still effectively left with 1.5 million species. I mean, we are scooping a small ladle full of water out of the top of a very full barrel. So what we're doing is, if you like, a demonstration project. we're learning how to do this sort of thing. We're learning how to build the resource that's needed to do a whole biota project. And we really want to make sure that when we collaborate with people in other jurisdictions outside Britain and Ireland, that we do it in a very sensible and open and respectful manner. So we want to develop the toolkits. And so to work with other jurisdictions, for example, low and middle income countries, we'd really be looking to try and establish indigenous, is that the right word, autochthonous, local capacity in identifying taxa, in selecting taxa for sequencing, and also in generating the sequence locally. I think with the new generations of sequencing instruments, it's now perfectly credible to set up a sequencing laboratory in what would previously been a very far-flung location. So our idea would be to build these collaborations to help people learn what they need to do and to support them in doing that. It's not going to be easy. We work inside international legal structures around the Convention for Biological Diversity, especially the Nagoya Protocols on access and benefit sharing, and also local jurisdictional procedures. That's one of the reasons why we decided to do our local biota, because we can legally, but we will be as ethical as we will be entirely ethical and entirely legal in what we do in the future. And that involves close collaboration with the people who actually, if you like, own or have guardianship of the biota in their space. So I think we ran a meeting recently, the Biodiversity Genomics 2020 meeting, and we had regional sessions in that meeting. One of them was from New Zealand and Australia. And I was really struck by the engagement of the Aotearoa New Zealand Genomics project with the local Maori traditional owners, and with respect to the very special legal situation that the land and the organisms that live in it and on it have in New Zealand. And I think that kind of attitude is something that's going to have to drive us through completing this, the super project, which is all 1.5 million.
Yeah, so the next question goes more into the technicalities. So if current taxonomies are based on the physical characteristics of a species, how do you predict a genomic-based taxonomy could disrupt this structure?
Yeah, this is fun. So I'm a zoologist at heart, and I always liked the groups of animals that had no place in current taxonomies. So I still work on nematodes, which in the taxonomy I grew up with at university were aschelminthes. And if you look at what aschelminthes are, they're just a group of animals which we don't really know where to put them. They also look a bit wormy, so we'll put them in the bag together. And so I've seen in just the last 20 years a complete revision and rearrangement of the animal tree of life, so all the phyla of animals, including things like whole phyla being relegated to being just suborders of other phyla. So real big rearrangements. That's all been driven by sequence. So I think what's going to happen with the sequences we get is, yes, we are going to rearrange the tree of life. We're going to realize new relationships, and that's going to reveal to us some of the pattern and process in evolution that we don't yet see, because we're going to realize that these two organisms, for example, have converged on the same solution to whatever life throws at them, even though they come from completely different parts of the tree. The other thing I think was really going to come out and is very exciting is this idea about how species form. So in many major taxa now, it's quite well understood that there's a lot of hybridization between species, and that the species boundaries or the species barriers are more porous than we might think. Now, what we classify as species now are actually hybrids or contain hybrid input. You think about humans and Neanderthals. There are bits of our genome, including bits of our genome that may predispose us to getting slightly worse outcomes with coronavirus, which are actually come from the Neanderthal branch of our ancestry. And I think that's going to be a really exciting thing to find. So not just the tree of life, but the trees of life, and understanding the major branches and the major bifurcations and the major radiations, but also how genes and bits of genomes have been swapped between taxa through life's radiation.
So we've got I think a question here that really strikes about the practicalities of this. And so the question is, what are the near-term practical ways that a naturalist, a biologist, an ecologist, someone who may work in the field today, but is not actually trained as a molecular geneticist, could really use these sequences?
Yeah, so I think I'd have to say that the direct practical application for a field naturalist at present is relatively low. So we wouldn't expect a field naturalist to have the sequence of a badger in their smartphone in order to be able to identify the badger that's coming out of the set in front of them. I think that's not what we're at. But I think what naturalists and people who manage natural ecosystems are going to be able to do in the near future is ask general questions about their ecosystem, about what species are here and how distinct are the populations of particular species that are found in this area I'm interested in from others. And that's only going to come if we have a good genomic foundation to lay that on. So I think for the naturalist now who's not a molecular biologist, I'd ask you to stay with us while we develop the tools. Your skills are absolutely needed. As I said in the talk, the Sanger is not a biodiversity organization. I can recognize a number of species, but we need to recognize, for example, 60,000 in Britain. And so we absolutely rely on the huge community of dedicated and enthusiastic naturalists to identify where these species are and help us understand the complexities of the data we find. And so I think in general, we found that the specialist communities, there are organizations, communities which are built up around particular groups of organisms. The Bees, Wasps and Ants Recording Society, who have very detailed and very erudite conversations about the ranges of particular species of wasps, or whether what's been seen is a new species or whether it's a subspecies, etc. And those people, I think, quite frequently get in detail the future benefit of this genomics process, because it's going to allow them to start thinking about, well, how does do the relationships between these species, which may be revealed by the DNA, result in these different morphotypes, and why are some species more endangered than others? Can we read that in the DNA? Have they always been rare, or did they used to be common, etc.? So I think watch this space for the naturalist who wants the genome sequence in their iPhone so that they can point their tricorder at something and see the genome. But in terms of immediately for people managing space, we're going to be generating libraries of DNA, which you can map environmentally sourced DNA or other things to tell you what's there and where they came from.
Yeah, so the last question, very shortly, how do you ensure that individuals selected are representative of the whole species genome, accounting for variation?
Yeah, we can't. And we are selecting individuals which are identified as clear members of the species by our expert taxonomists. But we're only sequencing, because we're sequencing from single individuals, we're only sequencing 2 haplotypes out of all, in a diploid, out of all the haplotypes, or out of all the alleles that are present in the population. So we can't, and we're not guaranteeing that this is the median representative of every species. What we can say is that my 2 sets of alleles in my 2 chromosomes, and everybody else who's listening, your 2 sets of alleles actually sample a lot of evolutionary history. So some of the alleles that you share with your neighbor, if you compare your alleles with those of your neighbor, some of those you share with a relatively recent ancestor, maybe only going back a couple of 100 years. Others you may not share with an ancestor until you've gone back hundreds of thousands of years. And so because of that, what they call coalescence science, because of that differential coalescence across the genome, just sequencing 1 pair of alleles actually gives us access to quite a lot of the history of an individual species. So we can say a lot about a species from just 1 genome. And this genome will act as a representative, but also it'll act as a foundation, which if you do want to chase the diversity of the particular species, this hoverfly, that dragonfly, this plant, you've actually got a reference genome against which to do it. So that's the next step.
Thank you. So we would like to thank Mark once again for his beautiful presentation and joining us for the Q&A session, and also the audience who joined this virtual session. There will be a seminar each month, so please look for announcements. Next month, speaker will be Dr. Dominik Kiulaski, who leads the Institute Parasites and Microbes program. His seminar will describe how they are using genomes to study and track infection diseases. Thank you for joining us, and we look forward to seeing you next month.